Hello, so today we're going to start our unit on molecular genetics by learning about the structure and function of DNA. So we're going to first begin by recapping the history behind DNA and kind of talking again about some of those scientists that really helped us grasp a greater understanding of DNA. The first scientist we're going to discuss is Frederick Griffith. And before we even write anything down on our notes about him, I just kind of want us to talk through his experiment so we can figure out what the heck is even going on here. So Frederick Griffith, he did an experiment where he took mice and he took two different strains of bacteria. One of the strains of bacteria was deadly and the other one was not. For the first experiment, which you can see over here on the left side of the picture, he took the non-deadly strain and injected it into the mouse, and the mouse lived. Big surprise, right? For the second trial, this one right here that you can see, he took the deadly strain of bacteria, injected it into the mouse, and the mouse died. No big surprise there. It's a deadly strain of bacteria. The mouse died. We get it. The next trial is where it gets a little bit more interesting. So he took this deadly strain of bacteria, the red one is the deadly one, and he heated it up so that the bacteria was no longer actually alive anymore. He injected the heat-killed deadly strain of bacteria into the mouse, and the mouse lived. It's because even though the bacteria is deadly, if it's not alive and if it's not active, it cannot actually harm the mouse. Still makes a lot of sense. And then this fourth trial is where things got a little crazy, and Griffith actually discovered something brand new that nobody had discovered before. So what he did is he took the non-deadly, this little blue strain right here, he took the non-deadly strain and he mixed it in with the heat-killed version of the deadly one. So right off the bat, nothing here is actually deadly to the mice in and of its own. The blue strain is the non-deadly stuff. We can see the mouse lived over in that trial. The red strain, that's heat killed, it didn't kill the mouse either. So what we should see is that the mouse lives after being injected with this combination. But what ended up happening is that the mouse still died. So this got Griffith kind of thinking, okay, what the heck is going on here? So to recap his results, a mouse that was injected with live non-deadly bacteria survived, a mouse injected with live deadly bacteria died, a mouse injected with the heat-killed deadly bacteria survived, and a mouse injected with the live non-deadly bacteria and the heat-killed deadly bacteria died. So what's truly going on here? Well, part of the heat-killed deadly bacteria was able to make it into the live non-deadly strain. Essentially what happened is even though the heat-killed bacteria is dead, it still contains the DNA that contains the proteins for making deadly bacteria. So the living bacteria that beforehand was non-deadly was able to take up some of the DNA from the deadly strain. This process is called transformation, and it's a change caused when cells take up foreign DNA. So what Griffith saw is that the non-deadly strain of bacteria, they were able to take up some of the the deadly bacteria's DNA and incorporate it into their own genetic code, making the previously non-deadly bacteria suddenly deadly. So this is what we really need to write down on your notes for Griffith. I want you guys to write down that Griffith showed that DNA could be transferred from organism to organism. And also, go ahead and write down the definition of transformation, that it's a change caused when cells take up foreign DNA. Our next scientist that we're going to be looking at is Erwin Chargaff. So Chargaff, he came up with our base pairing rules. So essentially there are 4 different nitrogen bases, which we will be taking a look at here momentarily as we dive further into the structure of DNA. But what Chargaff discovered is that whenever he looked at one single strand of DNA, the amount of the nitrogen base adenine always equaled the amount of the nitrogen base thymine. And then he also noticed that the amount of the nitrogen-based cytosine always was equivalent to that of the nitrogen-based guanine. So through this, he was able to determine that, well, if the amount of adenine is always the same as the amount of thymine, they're probably bonding together. The amount of cytosine is always the same as the amount of guanine, so those are probably bonding together. And whenever we take a closer look at the structure of DNA, you will see how the base pairing rules are essential to our understanding of the structure of DNA. The next group of scientists is Rosalind Franklin and Maurice Wilkins. So Franklin and Wilkins, they were the first ones to truly discover the structure of DNA. They didn't build the 3D structure, but they did take a picture of DNA using technology called X-ray crystallography. X-ray crystallography is a super fancy technology that takes fancy pictures of super molecular, in-depth things that we really don't need to go into right now. Basically, they took a picture of DNA, and that's what you need to know. Be able to recognize that X-ray crystallography is the technology that Franklin and Wilkins used to take a picture of DNA. Our last group of scientists is Watson and Crick. So Watson and Crick used Franklin and Wilkins' X-ray picture, and they built a 3D model of DNA. They had to go through some trial and error to figure out which 3D model was the true structure, I should say. And once they figured it out, they were able to share their findings with the world. But it's important to note that without the picture taken by Franklin and Wilkins, they would not have been able to have their starting point to be able to discover the structure of DNA, the 3D structure. So truly, it was all built upon each other, from Griffith to Chargaff to Franklin and Wilkins to Watson and Crick. All these scientists built on each other's ideas. Then in 1962, we had some Nobel Prize winners based on all of these amazing discoveries of DNA that were starting to come out in this time. We got Wilkins, who took the X-ray crystallography picture with Rosalind Franklin. We got Crick, we got Watson, but Rosalind Franklin never actually received a Nobel Prize for her work on DNA. Her partner did, Wilkins. See, there's him right here. There he is. But Rosalind Franklin, is missing. Yep. So now we're actually going to dive into the structure and function and learn what DNA is truly all about. So I want y'all to think back for a moment. Where is DNA located? We learned about where DNA is located whenever we learned about cells, and we learned that it's located in a different place for both eukaryotes and prokaryotes. So for eukaryotes, DNA is going to be found inside the nucleus, but prokaryotes do not have a nucleus, so the DNA is just found floating out in the cytoplasm. What type of organic molecule is DNA? Whenever we learned about biomolecules, we learned about the 4 biomolecules: carbohydrates, lipids, proteins, and nucleic acids. DNA is an example of a nucleic acid. We learned about DNA and RNA, and we're going to learn about RNA later in this unit, but for now, we're just going to start with DNA. The function of DNA is to store genetic information. So by genetic information, we mean all of the instructions that are used to make up every single protein that builds an organism. So it's basically the instructions for building all of the building blocks. So proteins, they're extremely important for organisms. They not only provide the structure, but they also are enzymes. So remember, enzymes are a specialized form of protein that helps speed up chemical reactions. Without enzymes inside your body, all of those reactions that are taking place every second of every day would not happen fast enough at a fast enough rate to keep you alive. So proteins are absolutely essential, and that's why DNA is such an important molecule, because it contains all of those instructions for making proteins. DNA is a polymer. Remember, a polymer is made up of 3 or more or many parts. So it's a long molecule, and it's made up of building blocks called nucleotides. Nucleotides are made up of a sugar, a phosphate, and a nitrogen base. I want you to make sure that you label those 3 things on your notes. Make sure to label the sugar, label the phosphate, and label the nitrogen base. On your diagram, number 1 is the phosphate. Number 2 is the sugar, and number 3 is referring to the nitrogen base. The sugar in DNA is deoxyribose. I want you to make a note of that somewhere on your notes page, because you should know that the sugar found in DNA is deoxyribose. The sugar found in RNA is ribose. And so those are 2 separate things, and I want you to know DNA stands for deoxyribonucleic acid, simply because it is a nucleic acid that contains the sugar deoxyribose. The phosphate group is simply just a phosphate, or it's a phosphorus atom with 4 oxygen atoms. You don't really need to know that much detail. Just know that it's usually represented by a little, like a little circle right here, with sometimes with a P inside of it, like that. And then you have your nitrogen base. So there's 4 different types of nitrogen bases, which we'll dive more into here in a second. The nitrogen base is the part that switches out in all of the nucleotides. The sugar and phosphate are always there, always the same. It's always deoxyribose sugar, always a phosphate group, but then it's the nitrogen base that changes. The 4 different types of nitrogen bases are adenine, thymine, cytosine, and guanine. Usually we represent those with A, T, C, and G, but it helps to know the full names as well. DNA is made up of 2 chains of nucleotides joined together by the nitrogen bases. Those 2 strands of DNA are going to twist together, almost like a twisted ladder. We call that twisted ladder structure a double helix. So the structure of DNA is a double helix with the backbone of DNA, so almost like the like sides of the ladder. It's made up of alternating sugar and phosphate molecules. You can see that over here in this diagram. We have a twisted ladder of DNA. It's going sideways right now, but the pink part right here, it's got PS, PS, PS. Those are alternating sugar and phosphate molecules, and they make up the sides of the DNA twisted ladder. And then the actual steps of the ladder, or the rungs of the ladder, are the nitrogen bases. I like this drawing because it kind of puts everything into a little bit more perspective. So we learned about cells, right? Here we have a cell. The nucleus is inside the cell. Inside the nucleus, you're going to have chromosomes. And then whenever you unravel chromosomes, you get the DNA in that double helix twisted ladder shape that we're going to be taking a look at today. So what chromosomes are is it is scrunched up, jumbled up, all twisted up DNA into an X shape. So once again, structure of DNA, we got sugar and phosphate backbone. We have the nitrogen base pairs that make up the steps of the ladder. And then this is kind of just the simplified version. And then the actual 3D structure is a twisted ladder like this, and it's called a double helix. Let's talk more about those base pairing rules. So if you think back to the beginning of this presentation, we talked about Erwin Chargaff, and he was the scientist that came up with our base pairing rules. What that means is that the nitrogen base adenine always, always, always pairs up with the nitrogen base thymine. A always pairs with T in DNA, and then cytosine always pairs with guanine. So if you take a look over at this picture on the side, you will see that every time we have a little adenine right here, we got a thymine on the other side of it. Every time we have a cytosine, we have a guanine on the other side of it. A pairs with T, C pairs with G, A pairs with T, so on and so forth. The nitrogen bases are held together by hydrogen bonds. Those hydrogen bonds are not very strong, which is a good thing because whenever we learn about how DNA replicates itself, we'll see that it breaks those hydrogen bonds in order to replicate. So the hydrogen bonds are not very strong, but they do hold those nitrogen base pairs together. Nitrogen bases that bond together are going to be called complementary bases. So, for example, adenine and thymine are complementary bases. Cytosine and guanine are complementary bases. Let's do a little bit of review, and you can, in the meantime, also label your diagram at the bottom of your notes. So first off, what is this little circle part on the end right over here? So that little part is always going to be your phosphate. The middle part right here is always going to be your sugar. And then these parts that make up the steps of the ladder are going to be the nitrogen bases. So as you can see, we have the phosphate sugar backbone here, alternating sugars and phosphates, and then we have our nitrogen bases in the middle. So the nitrogen base guanine is always going to pair up with cytosine. So this would be a cytosine on the other side right there. For the next nitrogen base, cytosine, C always pairs with G. So we're going to have a guanine on the other side. For the next one, T is always going to pair with A. The next one, same pattern, T always pairs with A. The next one, we have an A that pairs up with T. And then we have a C and that pairs up with G. So this pattern always stays the same. Those base pairing rules do not change for DNA. DNA does have two distinct ends of each strand. So there is an end that has a phosphate on it right up here. Remember, it's alternating phosphates and sugars. So there is an end that has a phosphate sticking off, and then there is an end that has a hydroxyl group sticking off. The hydroxyl is just this little OH right here. We've looked at it before whenever we learned about dehydration synthesis and hydrolysis. But it's easier to remember this little phosphate right here. So the way that we name these different ends of the DNA strands is the end that has a phosphate sticking off is called the 5' end of DNA. The end that has the hydroxyl group sticking off is called the 3' end. 5' and 3' are just terms, they're just names for the ends of these strands, so don't get tripped up by the primes. We're not actually talking about prime numbers or anything crazy like that. I always find it easier to remember that phosphate is the 5' end because phosphate and 5 both start with the same sound. So see if that can help you remember that phosphate sticking off indicates a 5' end, and the other side has to be the 3' end. Why don't you go ahead and take a minute to label the 5' and 3' ends of your DNA diagram at the bottom of your notes. It should, when you're done, match the diagram that I have up on the screen right now. Something you should notice about the 5' and 3' ends is that they run in opposite directions. If we look at the left side of DNA, it runs 5 at the top to 3 at the bottom. The other side goes 3 at the top to 5 at the bottom. So those two strands of DNA are running in opposite directions. We call that anti-parallel. So you can describe the two strands of DNA as anti-parallel because the two strands run in opposite directions. One goes 5 to 3 and the other one goes 3 to 5. Couple fun facts. If you wrote down all the bases in one cell, all the nitrogen bases, you would fill a stack of 1,000 phone books with A's, T's, C's, and G's. Kind of cool, right? If you unraveled all your chromosomes, so all those little X-shaped chromosomes in your cells, and laid out the DNA end to end, the strands would stretch from the Earth to the moon about 6,000 times. So much DNA in all of your cells. Cool. So last little bit of the notes, let's talk about kind of the bigger picture of it all. Here on the screen, we have a bacterium, we have some fungi, we have a cat, and we have Drake. So the question is, what do all these organisms have in common? On first glance, a bacteria, a fungus, a cat, and Drake don't really look like they have anything in common, right? They all act differently, they all look differently, they're all different organisms. However, the thing that unites them all is our genetic code. So all organisms use the same genetic code. We basically speak the same DNA language. Every single organism, from the bacterium all the way to Drake, are all made of A's, T's, C's, and G's. When you switch up the order of those nitrogen bases, you get a different organism. So two organisms that are more related, such as a cat and a tiger, they would have a closer genetic code than, let's say, a cat and a polar bear, right? So every single organism, you and me, cats, polar bears, Drake, bacteria, all made of A's, T's, C's, and G's. It's just the order of those bases that changes and results in a different organism.